Welcome to Genetics 101 and FOXP1 Syndrome. This is Jillian Carrick, International FOXP1 Foundation Genetic Counseling Intern. A diagnosis of FOXP1 syndrome in your family may bring up a range of powerful emotions, and those emotions may continue to change over time. And however you're feeling is completely valid, and we're glad that you found the FOXP1 Foundation, and we hear you. The goal of this resource is to help build a clear understanding of genetics concepts starting from the beginning and to become familiar with the FOXP1 gene, including its role in the body and the different kinds of genetic changes. We will also talk about the International FOXP1 Foundation initiatives and ways to get involved in research. And at the end, we will get some community questions answered. To use this resource, you can watch the video presentation with my voice recording, or you can flip through the presentation at your own pace, reading the text on each slide. You can also change the captions in the YouTube settings into your preferred language. Our bodies are made of trillions of cells, and each one of those cells contains the complete instruction manual for how the body works. This complete instruction manual, or DNA, is stored in the nucleus of each cell. And DNA is the full instruction manual for the body that is spelled out in a very long sequence of letters. Our DNA is organized into 23 pairs of chromosomes. And you can think of these chromosomes or these structures here on the right as books that make up this instruction manual. Inside of each one of these books, or chromosomes, are chapters, and we can think of these chapters as genes. So genes are those individual chapters within the books or chromosomes that make up the complete DNA instruction manual for the body. When we take a look inside the books or inside of our chromosomes, we will see many different chapters of sequences of that DNA organized into what are called genes. The FOXP1 gene is on chromosome 3. Since the FOXP1 gene is located on the shorter arm of chromosome 3, at position 13, we celebrate FOXP1 Awareness Day on March 13th. FOXP1 is one of 20,000 genes in the body, and they all work hand in hand. Each chromosome contains thousands of genes, but what does each gene do? Each gene provides instructions for how a specific part of the body grows or works. And depending on the type of the cell, specific genes are read. For example, eye cells will use genes that have instructions for developing the eyes and for vision. We all have the exact same set of genes. It's small differences within those genes that make each of us unique. And sometimes those differences within a gene can interfere with how the body interprets the instructions for the body contained within that gene. FOXP1 provides instructions for many parts of the body. Cells read the instructions in our gene chapters to make proteins, and these proteins carry out a job in the cell. And you can see in the diagram on the left here that each gene is made up of a sequence of DNA. And in the DNA alphabet, there are only 4 letters, and the cell can interpret that message as instructions to make a specific protein. Before the cell can make a protein, the cell has to make an abbreviated copy of that full gene chapter. We can think of this as taking handwritten notes from a full chapter in a textbook. Within the DNA sequence of a gene in the textbook, there are regions called exons and introns. Only the exons are included in the mRNA notes. Introns are not included, and we'll talk about those a little more later because they are important in some types of genetic variants. after the mRNA notes are written, the cell is then ready to make proteins. And proteins are built by reading the mRNA notes in groups of 3 letters, or codons. Each group of 3 letters is a code that matches specifically with a building block, and these building blocks are strung together to make a full protein. Now you may be wondering, what does FOXP1 do? And the FOXP1 gene builds a protein called the fork-head box protein P1. This protein is a type of protein called a transcription factor. It's special because it tells other genes when to be used by turning them on or off as needed. This process of turning instructions on or off is especially important in early development when many organ systems are being built at once. FOXP1 coordinates the development of these organ systems, such as the brain, the heart, the lungs, the esophagus, and even the immune system. And since FOXP1 is needed in many parts of the body, people with genetic changes in FOXP1 may have a spectrum of features. However, it's important to know that it's unlikely that one person would have all possible characteristics, and each person is unique. The brain is one area of the body where the FOXP1 gene and the FOXP1 protein are especially important. In the brain, FOXP1 helps support learning and behavior, speech and language, cell-to-cell communication within the brain, and the FOXP1 protein is active in specialized areas of the brain called the cortex, the thalamus, and the striatum, which help with movement, behavior, and verbal communication. And we want to remember that genetic variants in FOXP1 cause a spectrum of features, and not everyone is the same. To understand genetic changes in FOXP1, we have to remember that we have 2 copies of each of our chromosomes, including chromosome 3, where FOXP1 is located. Each chromosome has a copy of the FOXP1 gene. The body needs both of these FOXP1 copies to be functional to make enough of that FOXP1 protein for our cells. When just one copy of the FOXP1 has a spelling change in that sequence or is completely missing, this is enough to cause FOXP1 syndrome. And keep in mind that individuals with FOXP1 do have one functional copy of the FOXP1 gene, which still helps support important aspects of typical development. This is to provide an overview of the different types of genetic variants that we can see within the FOXP1 gene. We'll talk about each of these types of changes in further detail going forward. The first type of genetic change, or variant, is a spelling change in the FOXP1 gene. Remember, letters are read in groups of 3, and when one letter is changed, the meaning of the sentence or the protein product changes. As you can see in this example sentence, the cat ate the rat. If we change the R in rat to an H, this sentence now reads, the cat ate the hat, and has a different meaning. And as summarized in the graphic on the right, a spelling change in the FOXP1 gene can cause a change in the FOXP1 protein. This change in the protein may make the protein non-functional. There's another type of spelling change that can happen called a nonsense variant. And you'll see in the example sentence, the cat ate the rat, that this type of spelling change causes the sentence to end. So the cat ate the, and the sentence would end there. Some spelling changes create this end signal too early, and this cuts the instructions for the protein short, producing a shortened protein that usually can't do its job. The second type of variant that can happen is an insertion or deletion, which is exactly that. This happens when letters are inserted or deleted from the instructions. And when this happens, the grouping of 3 letters shifts, and every set of 3 letters after that gets read differently. This ends up with a protein that is completely changed from that point on and is often unable to do its original job. Not all types of genetic variants happen at the sequence level, where we're looking at the individual letters. Some genetic variants happen on a larger scale. In this case, you'll see on the left that sometimes the entire FOXP1 gene can be deleted. Or on the right, you can see that there can be missing pieces of chromosomes that may contain the FOXP1 gene and may contain other genes as well. The fifth type of genetic variants that we'll go over is called intronic variants. And you'll remember that when the cell takes those handwritten final notes from the gene chapter, those introns are removed. A change in a DNA intron can actually disrupt these notes of the mRNA, leading to notes that become scrambled when it comes time to make the protein. This can often translate to a protein that cannot do its job. Even though these variants look different from each other at that gene level, they can actually impact gene function and protein development in very similar ways. Scientists are starting to notice patterns between people who may have the same variant, but these patterns are not yet strong enough to predict an individual's outcome based on their variant alone. Stay tuned for ways that you can help scientists collect information to discover these patterns. We identify genetic variants and can tell the type of variant through genetic testing. There are several types of genetic tests, and the ones listed here on the left are tests that look at the spelling of the DNA sequence. The tests listed on the right look for structural changes or instances where pieces of whole chromosomes might be missing or duplicated and impact FOXP1. Each variant is given a classification based on whether or not that variant causes the gene to lose its function. Starting on the right side of this spectrum are pathogenic and likely pathogenic variants. And these are changes that we're confident do impact gene function and therefore impact overall health. Variants of uncertain significance lie in the middle of this spectrum and are changes that we can't tell yet whether they impact gene function, and they need further investigation to say one way or another. Not all changes in the gene cause differences in the body. This is called genetic variation, and it's part of being human. Variants that appear on genetic testing reports have been reviewed by teams of scientists, and these variants have been proven to cause health differences. However, knowing the genetic change alone or the variant alone cannot always predict what those exact health challenges may be. It's important to know that genetic does not always mean inherited. Some gene changes happen spontaneously or randomly by chance, very early in embryonic development or in the single egg or sperm cell that gives rise to an individual. These variants are called de novo variants, meaning that they were not inherited by either parent and happened spontaneously. Most times, the FOXP1 gene variants are de novo, but not every time. De novo variants happen due to random DNA replication errors as cells divide. In most individuals with FOXP1 syndrome, that genetic variant in the FOXP1 gene happens spontaneously, or a de novo variant, meaning that neither of their parents have a genetic change in the FOXP1 gene themselves. Variants are confirmed to be de novo oftentimes when parents' DNA is analyzed alongside the child's DNA in genetic testing. This technique in genetic testing is called trio-based genetic testing. This can identify uncommon instances where a parent carries a FOXP1 genetic variant without having signs or symptoms of FOXP1 themselves. I'd like to transition now to answering some community questions that were provided in a survey, and also to dive a little deeper on some of the topics that we already discussed. So the first question is, what if my trio genetic testing report says that I, a parent, have a FOXP1 variant, but I don't have any signs or symptoms of FOXP1 syndrome? And this is uncommon, but absolutely can happen. One explanation that we have for this scenario is a concept called mosaicism. And in genetics, mosaicism is a term used to describe a scenario where one portion of our cells may contain a genetic variant and the other portion of our cells do not have that variant. So, for example, some cells in the body may contain a variant in FOXP1, while other cells in the body are missing that variant, do not have any variants in the FOXP1 gene. And so when genetic testing is performed on parents, blood or saliva cells are collected, and a FOXP1 variant may exist in the blood or the saliva of an asymptomatic parent, but not in the brain of those parents. And this would explain how a parent's genetic testing would say that they have a FOXP1 variant, but the parent may not have FOXP1 syndrome. And so how does this happen? This happens from a rare event happening early in embryonic development. The embryo actually starts as one single cell and develops as that single cell divides over and over. If a cell acquires a variant along the way during this process of cell division, like this blue cell in the diagram, then the cells that are the product of that one blue cell dividing will then have that exact same genetic variant. So in our diagram here, if this blue cell picked up a genetic variant in the FOXP1 gene, every cell that is the product of that blue cell dividing will also contain that FOXP1 variant. And then the rest of our body is made up of those pink cells that didn't happen to pick up that variant in FOXP1. And now if the FOXP1 variant is seen in the reproductive cells of a parent, but not seen in the brain, this can explain how both a parent and a child would have the same FOXP1 variant, but only the child has symptoms of FOXP1 syndrome. Another possible explanation to answer this question of a parent that might be identified to have a FOXP1 variant is a concept in genetics called variable or incomplete penetrance. And this is a concept where a group of people, all with the same FOXP1 variant in every cell of their body, only a portion of those people might actually have FOXP1 syndrome. And this is a phenomenon that could happen in many different genetic conditions, and we don't fully understand how this happens. It might be a combination of the person's other genetic factors, their whole genetic makeup, but the cause of variable penetrance is largely unknown. And so, building off of these concepts of incomplete penetrance and mosaicism, I'd like to answer these two questions. Can FOXP1 syndrome be passed down to your child? And is it more likely to be inherited or de novo? And in those uncommon instances where we see incomplete penetrance or mosaicism at play, those families would have an asymptomatic parent that carries that FOXP1 variant, and therefore a child could have inherited that variant from a parent. But in the majority of families, FOXP1 syndrome is caused by a random chance event that happens in the single egg or single sperm cell, or happens very early in embryonic development, and this would be called a de novo variant or a sporadic variant. And it's really important to remember that either way, FOXP1 syndrome is not caused by anything a parent did or didn't do. We have absolutely no way of controlling which genetic variants we pass down to our children, and without genetic testing, unaffected parents would have no reason to suspect that they have a FOXP1 variant. A common question is, could there be any environmental cause for the variant during conception or pregnancy? And the answer is no. There is no known environmental exposure during conception or pregnancy that would cause a variant in FOXP1. Again, we want to remember that FOXP1 is not caused by anything a parent did or didn't do. Another common question is, how can I explain FOXP1 syndrome to someone who is not familiar with genetic conditions? And my suggestion would be, FOXP1 is a rare genetic condition that individuals are born with. It is caused by a change in one gene that can affect behavior, communication, and health. And the next question is, how do geneticists determine if a variant is pathogenic or benign? And so we all have small differences in our genes that help make each of us unique, and most of these changes do not impact our health in any way, and they are called benign variants. Geneticists will include variants that are considered to be pathogenic on genetic testing reports, since they are determined to likely affect health or the gene function. And so, to determine if a genetic variant is pathogenic, scientists will ask several questions about that specific variant. Some of these questions include, is that gene change or variant predicted to make a non-functional protein? Has that same variant been identified in other individuals with FOXP1 syndrome? And is that variant de novo, which would be a clue that it is more likely to be pathogenic, and many more other questions. Variants of uncertain significance also sometimes appear on genetic testing reports, and these are specific variants or gene changes that may have some evidence that suggests that they impact health, but not enough evidence to say for sure. And the classification of this variant may actually change over time as we learn more and more about that specific variant and how it may impact health. The next question is, can the FOXP1 gene be repaired? And it's really exciting to know that the International FOXP1 Foundation is actively funding early stage research to address this question. Multiple research studies are currently in progress, and research includes FOXP1 gene therapy and drug targets specifically to FOXP1 syndrome. However, it's important to know that these projects take a lot of time. These projects are currently in their foundational phases, and it takes time to develop safe and effective gene therapy for use in people. And it will require many more years of research before they move beyond the laboratory and cellular models that are currently undergoing testing. If you're interested in learning more, please scan the QR code or visit the URL listed on the bottom for all the updated FOXP1 research initiatives and more details on these projects. How can I contribute to FOXP1 syndrome research? The International FOXP1 Foundation provides several opportunities to engage in FOXP1 research. One way is by sharing information about your child that can actually help scientists uncover these patterns between FOXP1 variants and people with FOXP1 syndrome. This data sharing can influence the trajectory of future FOXP1 research efforts by allowing this data to be in one place to uncover these patterns and gaps in understanding. At LSU Health Sciences, they have launched a survey for families to participate in sharing information about their family member with FOXP1 as it relates to their immune health and infection history, as this is an area of FOXP1 syndrome that is less well characterized and understood. So by sharing some of your experiences with immune health and infection history, they can start to uncover patterns and understand what questions need to be asked about immune health and FOXP1 syndrome. At UCLA, Dr. Rajuda B. Wilson is studying the neurodevelopmental profile of FOXP1 syndrome. The goal of this study is to get a more complete neurodevelopmental profile of individuals with FOXP1 syndrome using neurodevelopmental assessments over time. These assessments are in person And there is opportunity to have remote assessments as well with Dr. Wilson's team. A unique part of this study is that Dr. Wilson's team will also share verbal feedback from these assessments for families to use and to better understand their child with FOXP1 syndrome. This assessment can be especially useful in sharing with your FOXP1 children's clinical team. It can also help better inform things like occupational therapy and is a really unique part of this study. For a full updated list of opportunities to participate in FOXP1 research, scan the QR code below or visit the URL. If at the end of this presentation you have questions specific to your family, a genetic counselor in your area can help and can continue this conversation. To find a genetic counselor in the US or Canada, you can visit findageneticcounselor.nsgc.org, or you can scan the QR code and it'll take you there. Thank you so much for listening and taking the time to learn more about the foundations of genetics and FOXP1.